as a physicist, and then discovered biology, and sort of got drawn into also experimental work, right? So then she was sort of out of the theoretical domain into the real world, and since then she's been investigating hippocampus, oscillations in the brain, and its broader relevance, but today she will focus very much on her work on the hippocampus, also as part of the EpiSense extravaganza we are exposed to. So Liset, it's really great that you're here. I met Liset when we were reviewing projects in Paris, and I was really impressed by her fresh attitude on sort of evaluation of projects in neuroscience. That's why I thought it would be really great for us to have her here and also share this freshness with all of us. So Liset, great that you're here. Thank you very much.
Okay. Okay, so thank you all for the introduction and for the invitation, and it has been 2 fantastic days. I have the opportunity to discuss fresh ideas in neuroscience, and actually this is about. So I will like to bring a new perspective about hippocampal function, and for that we need to recall what is brain about. So okay, so since Berger obtained the first electroencephalographic recording from the human brain, we soon realized the complexity of the activity that the organ, the brain as an organ, generates as compared with the simpler heartbeat. Actually, since then, we have been struggling with how to interpret these highly variable and non-stationary signals, apparently non-stationary signals. This tells us that the brain basically produces a wide range of oscillations from very slow to very fast, and some of those oscillations in the different scales are connected with different states, like slow oscillation during a slow wave sleep or more faster oscillation during attention. Now, if you consider the dominant frequency of these bands that are preserved along a species, therefore that no matter of the size of the brain, of the organs, the main frequency is preserved, this tells you that basically the mechanisms responsible for those oscillations are retained in all these brains, and basically these are the microcircuits. Not only the main frequency of an oscillatory component of the brain, of the activity that the brain produces matter, but also matter how different oscillations interact. And we know today that the slow and fast rhythms are coupled, and they are coupled in meaningful ways that connect with the cognitive function. For instance, theta gamma coupling is a major component of these interacting dynamics, like oscillations in the theta wave. The amplitude of high frequency oscillation like gamma are modulated by the phase of theta, and that is particularly meaningful for regions along the temporal in the hippocampus and the temporal lobe, and some other regions in the human brain, for which they exhibit this particularly theta gamma interaction during some memory tasks. Like for instance, in these experiments in which when the person was asked to hit between different scenes, the number of correct matches were correlated with an appropriate phase relationship between the amplitude of gamma and the phase of theta. However, when those mismatch, cases in which the hits were incorrect, they were actually associated with an opposite phase relationship. Basically, when there was no misses of the categories, there were no clear oscillatory components. So the work of many researchers has been trying to put forward that this level of interaction between oscillations can be signaling or demonstrating this cognitive ability, and basically the way in which different regions interact. I will try to talk about all these from the perspective of oscillations and how these oscillations are generated by the circuits. So we have been hearing about plate cells, grid cells. So how these cells emerge and how they, the activity from those cells in a way, can be explained by basic principles of how they are connected, which other neurons are sending inputs to these cells in the different structures. Now, a suitable structure to understand this relationship between rhythm and cognition is hippocampus in rodents. And basically, we are more or less all in this conceptual framework, on which we know that when a rodent explores the environment, what you record from the brain of that rodent, from the hippocampus of that rat, is theta. And basically, the firing of the place cells along CA1, for instance, are establishing a particular relationship, the firing, the single spikes, along this theta. We have heard about this theta phase precession. Interestingly, when the animal make a round between different, from left to right, for instance, that is traversing the different place fields, the order in which these cells are recruited is follow a particular relationship, which is temporal and spatial. Interestingly, when the animal gets to sleep and you have these high frequency oscillations which are waves that we will talk about later, ripples, basically what you can see during this short period of time, which lasts no more than 100 milliseconds, is that the sequences that were experienced by the animal during the exploration replay. They can replay in different orders, reverse or not. There are questions here that we still don't understand, but basically there is this replay selectivity there. So we call about the online mode of the hippocampus, which basically is when it's recollecting the information during exploratory experience. And we call about this offline mode, which is running when the animal is offline from the task, either immobile or getting asleep. And the oscillations that determine the relationship between the firing and the activity of the brain in the online and the offline mode are quite separate in the frequency band. Now, what are the microcircuit mechanisms? What is the microcircuit structure? So for that, probably we need to think about basic work. And basically, the father of neuroscience, Santiago Ramón y Cajal, put these arrows in his drawings. Evidently, he didn't see those arrows in a microscope, but basically what he tried to mean was that there was some directionality in which the circuits and the activity was flowing through this circuit, through different neurons. And basically, this gives to one of the most important concepts in hippocampal research or hippocampal physiology, which is the trisynaptic circuits. Trisynaptic circuits start in the entorhinal cortex, make a synapse in the granular cell layer, which then send activity through the axon to MOSI fibers to contact CA3 that send back projection to CA1. Trying to think about that and trying to put these trisynaptic circuits into the framework, we know that the medial entorhinal cortex has cells that code for position in space, so that the cells can inform about where we are in the space. Whereas cells in the more lateral cortex have may signal, well, the category of an item, what? We were discussing along these days about these different coding in the lateral and in the medial entorhinal cortex. Now, this activity converges through the trisynaptic circuits and eventually map into the granular cell layer, where the cells are connected sparsely and they fire very poorly. And that generates a pattern separation of the different items that are eventually represented in the entorhinal cortex. Because the granular cells send the axons to CA3, and CA3 is somewhat recurrently connected, like an associative networks, that generate a kind of attractor dynamics, and therefore you complete the pattern, so the pattern can be completed there. And because of the trisynaptic circuit said that CA3 then project to CA1, then you can there bind the two information that we're gathering here, because these layers were projected into the dentatic arrows. Now, the problem is that we know that this is very simple. And we know that there are other components in cognition which are also represented in the activity of the cell, which are in these regions. For instance, we know that the time when something happens, is somehow represented, somehow has to be stored in this network. We also know that the way in which all these circuits, these different cell types along the deep and superficial layers of the internal cortex project into the hippocampus is not that straightforward. Actually, we know that layer 2 and layer 3 project differently. Layer 2 projects into the dentatic gyros, or the layer 3 project into directly through the temporal harmonic pathway into the CA1 distal layers. So somehow these where, when, and what items should be integrated in the CA1, so that CA1 is generating a more complex and contextual representation of the information. And basically, we may know when happened and put that in context of other memories we had, and eventually give us like a kind of more complex memory capability. Now, the way in which this happened in the microcircuit is not by miracle. It is not that we said there are grid cells in the internal cortex that project to CA1, and then we should have some convergence there, and therefore we have some plate field or not a plate field. Actually, we need to know much more about the circuits. And one basic component of this complexity basically is regarding how the different cell types in the CA1 are organized and are exposed to those inputs that are coming into the hippocampus. For instance, pyramidal cells, they can collect activity from the basal dendrites in the orient alveolus and the apical dendrites. So the temporal harmonic pathway that is projecting from the internal cortex is reaching the very long distance, the very distal portion of the CA1. Therefore, these dendrites here are collecting information from the internal cortex. And you know that the distance matters. So how much these inputs have to travel till they go to the soma so that the soma fires. So those are more distal than those other inputs that are coming from the CA3, from the chapter collaterals, that contact with CA1 very close into the dendritic domains. On top of that, you have a wide variety of interneurons, GABAergic cells. Those are glutamatergic cells, the cells that fire and project to other regions. But interneurons, GABAergic interneurons, they are quite diverse. Some of them have their dendrites concentrated in a particular layer. Therefore, these cells here will never be activated by the chakra collateral. This means these cells here will be only responding to entorhinal inputs. However, those cells here, which are perisomatic cells, they have very poor dendrites coming into the stratum lacunosum moleculare, which is where the entorhinal inputs are arriving. Therefore, these cells, these interneurons, which are PD basket cells, I will talk about them, they poorly respond to the entorhinal. They shouldn't. There is not a channel to speak with. So it is not that the activity is converging in the hippocampus, it's what cell type has been contacted. So that schema has been put forward by the work of Somogyi and Klausberger. The only way to try to understand that is to record individual cells and classify the individual cells and try to understand how these individual cells are firing and participating and being activated during this activity. So basically, the CA1 circuit is composed of these pyramidal cells and this diversity of interneurons that can be activated by different inputs coming from the entorhinal cortex or from subcortical areas, the septum, on the CA3. And the way in which these different cell types fire during theta, we can take this kind of fixed picture of what is going on at the average level. If we take all of theta cycles, for instance, and we collect all of them, we know that pyramidal cells, they fire in the throat of theta. We will learn later that there is some diversity here. But PP basket cells, which are GABAergic cells that contact pyramidal cells along the somatodendritic region exclusively, those fire typically in the falling phase of theta. Whereas CCK basket cell, which is another cell type that innervate perisomatically pyramidal cells, but express a different marker, those fire before PD basket cells, and actually they fire more close to the theta peak, and so on. Other cell types have different firing preferences along this. Now, during ripples, those those players are activated differentially. For instance, pyramidal cells, they fire very clearly during ripples, together with PB basket cells, but not much with CCK basket cells. CCK basket cells. Whereas, for instance, axoxonic cells, which are interneurons that innervate the action initial segment, they are transiently inhibited, as well as OLM cells, which are a very important cell type because they are here in the orients, or they have dendrites not exposed to neither the CA3 nor the dentati, neither to a Schaffer collateral, neither to the internal cortex, but they are preferentially recruited by pyramidal cells. While these cells which project distally, they are typically inhibited during ripples, and actually they fire in phase with pyramidal cells. So that relationship is pretty much fixed, and we can make this taxonomy and classify cell types and know about the spatiotemporal relationship they have with the theta. Okay, but then we have this interaction between theta and gamma, and eventually during this interaction between theta and gamma, we also have a kind of different relationship between the way in which different cells are firing. Now, what can we is this picture fixed, or is evolving, or how does it connect with those questions we were discussing? For instance, how does it connect with that hypothesis that encoding and retrieval is occurring in different phases of theta? How does it connect with the fact that the cells are having this phase precession dynamics? Now, we can learn a lot if we look at how this happens. So these are recordings, laminar recordings along the different layers of a freely moving rat during theta activity. The animal is exploring the arena, is solving a memory task, actually. And you can see this relationship between theta and gamma very clearly. Now, if you record these with electrodes that are targeting the cell layers, basically tetrodes, what you can see is that at this level you have this relationship between the theta phase and then gamma. So that gamma is occurring in the falling phase of theta. And then you have that depolarization, and then so on. But if you look at cycle by cycle, you see that there are some variability here. And actually, on some points, you cannot really identify theta cycles with electrodes at the stratus pyramidale. But if you are recording simultaneously, as you can do with this probe, in which you record from several electrodes along the track, you see that at the level of the stratum lacunosum moleculare, you have the same kind of relationship, although it is reversed, because theta is reversed at that point. So you have this gamma activity occurring here in the rising phase, which is going in the same direction that the gamma activity occurring in the falling phase. And actually, you see that some of those cycles in which you cannot clearly define the oscillations at the level of the somatic recordings, you can clearly have some of these oscillations at the level of the stratum lacunosum moleculare. So this means that the flowing of the current is, the currents are flowing through the structure. Now, if you, one of the important concepts that derive from this consideration of different oscillatory bands channeling different lines of information comes from also the discovery by Laura Colgin in the Moser labs about this relationship between a wide range of gamma activity along the phase of theta. And basically what they proposed was that the fast gamma was, they record more close to the theta throw. Remember, these are recording with tetrodes occurring at these layers. Were occurring at this particular phase of theta, whereas the slow components were being distributing along the theta phase and basically concentrates in the falling phase. Now, if you try to understand whether this makes sense and how to make sense physiologically to those particular relationships, you can try to extract information from these multi-site recordings. Now, you look at the theta gamma phase relationship near the stratus pyramidale, you see that this component here, which is a kind of between 40 and 90 Hertz, are occurring near the peak of theta, and then it's going following by this slow component, which is between 30 and 40 Hz, that is occurring in the falling phase of theta. Whereas a faster component, as described, was more associated with this shift here occurring between 90 Hz and 150 Hz, and that is most aligned into the theta throw. But just there, you also have the contribution of multi-unit activity. So it's quite difficult to separate here at this area that different components, but you still have some high frequency components coming at this. But if you look at what is going on at the electrode, at the stratus radiatum, you see that at that level, you have a dominant contribution at the falling phase. That extends actually along the scale from the 30 to probably 80 Hz around here. And if you go to the electrode in the stratum lacunoso moleculare, now you will see that actually the component is become more faster as compared with that one, which is more here. But that component, faster component here, is more aligned to the peak. Now, this recording may have a lot of volume conducting effect. So you need to try to extract the generators. And there is a procedure in doing that, which is called current source density. And current source density is the second spatial derivative of the LFT signal. So if you do that, you can see the regions where there is currents, which are sinks, that are flowing into the cells, and therefore are depolarizing the cell. And you can follow that currents along the theta phase and along the area. So basically, the signal around the stratus radiatum and around the stratum lacunoso molecularis tells you that the specificity of these two gammas here are phase related with the incomes or the inputs coming to this layer, to this layer. So that at the stratum lacunoso moleculare, the maximum inputs, the sinks, occur at the theta peak. And here is where you can detect these high frequency component, more intermediate gamma oscillations. Whereas in the regions of the phase where you detect the inputs coming from the shaft recolaterals is where you can see these more slower components. Why? Basically because, as we were discussing, the internal inputs are coming into the theta peak is there where these CCK basket cell and axoaxonic cells are being seen to be maximally firing. So those cells innervate the perisomatic region of pyramidal cells and the axoaxonic region of pyramidal cell, and therefore, if these cells are firing here, those guys will not be firing because they are massively inhibited near the perisomatic region. During the falling phase of theta, where CA3 inputs are arriving, they activate typically PV basket cell because, as I said, those dendrites are there to be activated, and therefore these cells are innervating again into the pyramidal cells, and therefore they can control where they're firing. So as a result, it's very hard that a pyramidal cell will be firing along these phases. The only places where the pyramid cell will be to fire is just here, because basically the type of interneurons which are activated here, like OLM cells, are projecting distally here. And those cells, OLM, are activated precisely by pyramidal cells. So that is the fixed picture. But things become much more complicated. If you know that medial internal cortex project to the proximal CA1, which basically means that they project more close to the CA3, to the border with CA3, whereas lateral internal cortex project more to the distal CA1, which basically means that they're more close to the subiculum. So therefore, you should expect that if I am receiving much more of these inputs close to the distal border and much more of those ones close to the proximal border, I may have an expression from the proximal to the distal of those oscillations, because of those inputs put in motion those different type of interneurons, basically. Those guys here should be responsible for the very fast components that are occurring here, basically because these gamma activity is reflecting this excitatory and inhibitory activity. So actually what you see is that for the 30 to 60 Hertz band, which is basically aligned with the Schaffer component, a Schaffer input, is stronger at proximal than at distal, where the reverse occurs for the more faster component. So that makes another level of complexity here in the way in which this circuit is established is proximo-distal. Then there is still another complexity, which is the fact that if an animal is, we are talking about coding for a space, this shouldn't be fixed, because if it is fixed, it's rigid, and you cannot decode any information. But basically, because we know that when the animal is running along the place field, you have this cell precession relationship. Therefore, the phase in which a pyramidal cell is firing when it is in the middle of the place field, or when it is ahead of the place field or back after the place field, is evolving through this cycle. So that means that a pyramidal cell will be firing along the faces with a maximal firing rate occurring here, but what is going on just here and there is meaningful for those other cells. If you look at how these different gamma boots are organized along the theta, you see that variability. Now, if we could learn enough from the system to decode the different memory items that are coming on here, this is exactly the online mode of the hippocampus. So whatever the rat is thinking, whatever the rat is doing, is just encoded in this particular oscillation that is occurring along the faces. Yes?
So on the previous slide, pyramidal cells were mainly, the distribution of spikes in pyramidal cells were logged through the peak of theta?
Yes, basically, as we saw, if you take the grand average,
Right.
so basically they fire here in the peak of theta. But you have firing occurring also at the other phases. It is just that this is more the most common phase for a pyramidal cell. And the explanation for that is precisely the circuit. And as I said, if you take the whole picture fixed, this is what is going on here. Because basically here, these axonics are inhibited strongly, the action in iliac segment, and this cell, even if they fire, will not travel. And these perisomatic cells are innervating all these basket cells are innervating perisomatic region in pyramidal cells, and therefore they will be also, okay? So now, what determines the phase firing of the, yes, of course, yeah.
You analyze CA1.
Yes.
But CA3 will show a comparable kind of dynamic.
Yeah,
so it's not that the spikes from CA3 that are driving CA1 are coming in as a random stream. They already are strongly segmented in time,
yes.
right?
Yes.
So that might in some sense offset the dynamics that you now sketch, because this is purely defined looking at the intrinsic properties of CA1, but this can in turn be entrained by the CA3 rhythm.
Of course, actually,
right?
Yeah, of course. Actually, if you look at CA3 is firing here. And the different layers of the entorhinal cortex are firing along these phases. So yeah, those reflect the inputs.
But I mean, because of the temporal structuring of CA3 responses, the actual dynamics of CA1 might actually be broader in some sense than this would suggest, because entorhinal comes in on the rather distal parts of the dendrite, so it's not a very dominant driver, and we already have a very strictly segmented CA3 response, which is much sparser than what the dentate would tell it.
You will see how dominant a distal driver could be.
Excellent.
Don't take that for, I mean, if I did my job well, I bring the idea that you should not read this cartoon as a fixed picture, but you need to understand what is important about the cartoon is where is the maximal, the preferred phase of the different cell types, where does it occur, but that there will be deviation from that, and we need to understand what is meaningful. So, okay?
Very good. Yeah.
Okay, any other question? Yeah.
Well, the phase procession, you know, we also see it in CA3 and in entorhinal cortex. So it's not clear where it's generated.
No. I am not meaning.
Maybe this is the same point. Yeah.
Yes, I am not meaning that this phenomena is generated. I just mean that there is an evolution of the firing phase of a particular cell, and that we should understand why a particular cell fire at the different, because that is important. I mean, so is that important where the cell fire? Yes, because it is important with respect to the other cells and what ensembles are being formed along the time, okay? So what determines the phase firing selectivity of this CA1 cell? Now, as I said, I mean, this is very laborious, you know, because if you come with tetrodes or multi-site recording and you have many cells, and thanks to that, we have been able to understand, to decode part of this complex representation. But it's laborious because we now know that those details of the cell types are important, and these force us to record a cell. So it's one brain, one question, one cell to try to find the answers, okay? So you will see along the presentation the many ways we do that, but one of these ways is head restraining animals. So we take a mouse or rats and we have them to feel comfortable having head restrained. They run in a cylinder. I don't show here, but there is a virtual reality task here, but that doesn't matter at this point. Now, if you record, we combine silicon probe recordings so that we can record 16 channels along layers, as I said, and together with a glass pipette so that we can record from a single cell. Now, if we do that, for instance, we can record a single cell, we can stain the cell, label the cell, and therefore identify the cell within the CA1 region, and therefore we can run the identification of the cell. Now, when you record, for instance, this CA1 cell is pretty much what you expected, actually. This is the theta phase at the pyramidal layer and the theta phase at the stratum lacunoso moleculare. By the way, look at the different signal-to-noise ratio that you have in these two different layers. I mean this point because if you are recording with tetrodes here, sometimes it's quite difficult to identify theta cycles. Whereas with the multi-site recordings, you can record theta cycles here, and then goes there and identify the associated components. But anyway, so basically, we always take as a reference the stratus pyramidale because this is the way to understand also tetrodes recording. So this particular cell here was doing what it's supposed to do. So the cell was firing at the theta throw. But then we record from other cells. And here is one cell which is doing just the opposite. So that cell actually is pyramidal cell and is firing actually at the theta peak. So that dogma is not that correct. Well, maybe this is a minority, maybe what can explain that differences? The three synaptic circuits we started talking with is no longer valid to try to understand that. And actually, this is a picture, I'm sorry that it is in Spanish, it's the PhD thesis of one of my brilliant, more brilliant students, Manuel Valero. And basically what this picture summarizes is what we know today about the different cell types that project to the hippocampus and how they are connected with the different hippocampal cells. And I will not have the time to go through over this, but basically, if you think about the internal cortex, we were talking about grid cells, border cells, boundary vector cells, head direction cells. Nobody knows really what identity they have. And if you go to the internal cortex, you see that in layer 2, for instance, you have this wide dispersion of cells that basically are composed of stellate cells. But there are some islands in which you can record from cells which express cal binding at double FS1, which is a marker of CA1, never expressed in these guys here. Those cells are called island cells. Those cells are called ocean cells. And these cells has been shown to project directly into these interneurons that inhibit pyramidal cells, whereas those cells generate the perforant pathway and project directly into the isolate and also into CA3 and CA2. Therefore, CA3 and CA2 and CA1 are not receiving from the cellular population in the internal cortex. So if you go to the layer 3, which compose the temporal monic pathway, we also know that there are some cells here which express different markers that actually, apart from some of them projected directly into the layer 3, they also contact directly here the distal dendrites of the CA1 pyramidal cells. Therefore, the picture is really much more complicated. The trisynaptic circuit is no longer valid to try to understand what is going on. We need to update the hippocampal physiology to try to understand cognition. We need that because the only way to try to explain how these different cell types are organized, generated here in the terminal cortex and converging into CA1 is by trying to crack this code from the most complicated cell type specific point of view. Remember, we still don't understand how time is represented. So in trying to go a bit about this, I would like to, one way to try to start understanding this puzzle is try to look for a direct connection. So what can survive from these trisynaptic circuits? Well, basically what survives is that we know that there is a direct connection from CA3 to CA1. At least we know that. This was by the work of Schaffer. Actually, those arrows that Cajal plays were inspired in a way by the work of this Hungarian researcher. So Schaffer noted that collateral from what he called region inferior, which were CA3, project to region superior, which was CA1. He also noted that there were layers here comprised of large and small pyramidal cells. So that is a pure interhippocampal pathway. So what this pure interhippocampal pathway can tell us about this circuit organization and whether it can provide another way to understand this puzzling representation. Now, the classical physiology of the trisynaptic pathway tells you that if you place electrodes in CA3 and you stimulate CA3 and you record along CA1, you will see a kind of response like this, which is basically at the apical dendrites. You will see a negative field, which is associated with a sink that represent the inputs coming into the dendrites of these CA1 cells. Around the soma, you will see this source associated with a spike, because if you make this stimulation strong enough, you will make all cells firing together at that very same time, and therefore you will have this population spike here, which is reflecting the firing, very precise firing of all these cells. So that tells you that CA3 connect directly to CA1, and there are spontaneous activity, as we were discussing, that reflect that. One of these is Char wave ripples. Char waves are initiated in CA3 because CA3 cells fire before the ripple on the Char wave is recorded in CA1. This is a simultaneous recording along the layers, the stratus radiarum with the Char wave, the stratus pyramidale with the ripple, and here is an intracellular recording of a CA3 cell, which is just here. So you see that that cell fire just before the peak of the ripple, the peak of the Char wave is recorded in the stratus radiarum of CA3. That, okay, so that actually, if you record with different layers along here, with different channels, electrodes along these layers, you see that Char wave that reflect the incoming inputs from CA3, the build-up that occur with those cells are start firing, and then this kind of high frequency oscillation that occur near the stratus pyramidale, which is actually the ripple. So the discharges from CA3 cells over the apical dendrites of CA1 are the main components of these Char waves, but there is a complex local field response, the Char wave at the radiarum and ripples at the stratus pyramidale. Now, what you should expect if I record from CA1 cell is basically a depolarization, because they are receiving this discharge from CA3. So an intracellular recording of a CA1 cell showed that. But then we found other cells that actually, during Char wave ripples, during these Char waves associated with the ripples, you didn't see a clear depolarization as this. For instance, some of those can even fire, but those cells were typically inhibited. So maybe it's a problem of the resting membrane potential. So we take the membrane potential and we move the membrane potential of the cell, and we saw that for those cells here, even if we were moving the membrane potential, they typically never like to be inhibited. They never show a negative response associated with the char wave ripple, or char wave peak. For those, what we found was that they were typically inhibited, but if we somehow take them more hyperpolarized, we reverse that. And actually, the resting membrane potential of these two cells is pretty much the same. So this tells you that at the resting membrane potential, some cells have a depolarizing drive, some other cells have a hyperpolarizing drive. More heterogeneity, more variability. So how could it be that we can emerge with a single framework? So we stain those cells, we label those cells, and we try to localize those cells. And what we found was that those cells, pyramidal cells that were basically depolarized, they tend to be more close to the radiatum. But those cells that were more basically hyperpolarized, they tend to be more far away from the radiatum. And actually, if we estimate the main depolarizing drive, the cell field or hyperpolarizing drive, basically at the resting membrane potential, how much was this drive, positive or negative? And we plot that with the distance to radiatum, the border to radiatum defined from all the cells. What we saw was a trend. And actually, if you look for Cal binding, which is a marker, Cal binding was expressed just in these cell types here, close to the border to radiatum, but not those guys here. So actually, these cells are early born cells. Those are the cells that are firstly generated along embryonic development. And then is coming these cells. And actually, because of that, well, the hippocampus is inverted. Those are called deep cells. Those are superficial cells. Basically, the same embryonic development programs that determine, well, more or less, not exactly the same, because hippocampus and the neocortex diverge, but those cells are differently generated. And it seems so that I have a quite antagonist relationship between them. Now, if you look at how, for these cells which are superficial, you look at the instantaneous relationship between every char wave and the sink associated to every char wave. We have char wave of different amplitudes. Yes?
Do you find the difference in the spike form of these two types of cells?
We found, not in the intrinsic spikes, we found differences in the firing rate during the events. You will see this now. So, as I said, if you take now, let's go again to the variability. So we have different char waves, different amplitudes. So some are larger, some are smaller. And we collect the intensity of the inputs that are associated, the larger one with bigger ones, the lower with, and we collect that, and we try to see whether there is a relationship with how much depolarization or hyperpolarization the cell has. You have a very strong relationship, meaning that the smaller this sink, the lower the depolarization. The larger the sink, the more. And actually, that happened for superficial, but not for deep cells. And it was pretty significative in a majority of superficial cells. Some cells didn't find that, but not for any deep cells. So this means that those cells, either they are not receiving from CA3, I tell you, no, they are receiving from CA3. Either there is another component, couple. You can guess what? Inhibition. So superficial cells are more responsive to CA3 inputs. They are innervated by CA3 both in superficial and deep, but they are more responsive. Actually, and this goes to the point. So in this variability, event after event, consecutive event, you have some events which are more depolarized, some events which are less, and that correlates with how much the cell is firing. Also, some events for the deep cells which are more hyperpolarized, some events that are composed of some depolarizing components, so that is a variability. But if you take an average and you look at the firing rate during ripples, what you see again is a trend with the distance to radiatum. So again, those cells here tend to fire much more than those cells there. Now,
but wait, before we go there.
Yeah.
So should we think about this as an intrinsic property of these cells or as a circuit property?
It's a circuit property. Intrinsic property cannot explain that.
Okay, so you're saying that these cells closer to the border are more tightly coupled to inhibitory neurons, essentially.
Yeah.
But that would suggest that also in their response profiles, that was what Diogo was asking earlier, they should look different. Like their response onset might be different.
Yes, of
or
course.
Their response
I'm
offset.
coming with all this.
Okay, good.
I'm coming with all this. So, but intracellular recordings, you didn't ask, but those were obtaining in anesthetized animals. It's impossible to obtain these intracellular recordings with such a level of fidelity. So far, we are trying to get this from heterostrained. With patch recording can be done, but patch will dialyze the cell and we will not be able to recover precisely the entity of the cell. Anyway, question is whether this happens again in freely moving. And then, so we obtain juxtacellular recording from freely moving rats. And for that, we work with, basically we now work with 2 different drives. So we use micro drives that allow us to bring the pipette close to the cell. We do this with a manual micro drive designed by Robert Aberking as a collaborator, long collaborator of us. We also have this motor drive, which actually was designed for intracellular recordings in birds by Michael Long, and is had been applied to recording in hippocampus both by the lab of Bushaki and ourselves. And you can bring a pipette close into that cell, record the cell, see the cell firing, build an autocorrelogram of the cell, and then eventually you can pass a current. And when you pass the current, you can drive the cell firing, and the cell will be stained because with the current, you electroporate the cell with the biositin. And therefore, after, you can identify the cell. Now, using that, we asked whether we can see this deep superficial in the freely moving animal. Now, you can think on all the contingencies and difficulties in seeing that. Differences between the states, differences between place fields. So can we be able to identify those variability? So basically, for instance, we record in cells and we try to identify differences between ripples, well, sharp wave. Evidently, these are juxtacellular recordings, which means that we are close to the stratus pyramidale. We don't have the multi-site recordings here. So we identify sharp wave ripples by the events. So recording during sleep, and the very same cell recording in awake condition, and we try to see whether there was some differences. And actually, we saw interesting differences. Some difference were in the direction of firing rate becoming higher for a cell participating in awake, but we also saw some cells that actually were modulated the reverse. We can't explain that variability at this moment, but that should be meaningful. But anyway, what we found was that in general, the sharp wave, the participation of the cell of sharp waves were higher in the wave ripples than the sleep ripples. So this could complicate interpretation. Second thing, as
about the proportion of these 2 populations, or the 2?
Which one? The 2. The more or less roughly, we got like almost half, but for in this database is where 2 cells over 1, 2, 3, 4, but we are collecting more now and we see them differently. So that has to be also with the deep superficial position, but at that moment we didn't have enough information. Now we have more, but we can discuss about this later. Okay. Anyway, so then that depends where the sharp wave is occurring, because if the rat is moving around, you may have an influence of the firing rate. Evidently, if you have a placed cell and the rat is having his snap here, so probably we will have a higher participation than if the rat is sitting around here or there. So, and actually, if you look at the relationship between the number of places a rat was moving around in this freely moving recording, you see that there is a strong variation. And after that, can we still capture a deep superficial gradient in these conditions? Yes, we can. And actually, we could identify the clear trend in the sharp wave participation and the firing rate during sharp waves for all cells, depending on the distance to the border to radiarum. Some cells we were tested for calbinding, whether they were positive or negative. Some other we couldn't test it, but all of them together, they exhibit this relationship. So hippocampal pyramidal cells are functionally organized in deep and superficial layers, and that seems to explain some part of the variability. So how does it relate what all these questions we were putting forward? What are the microcircuit determinants? So you may guess, or start guessing, that how inhibitory cells are innervating these deep and superficial layers should be different. And actually, what we did, we used a marker of the CA1, which is FS1, that mark all the cells. And then we look for Jeffrin, which is a protein that is expressed in the synaptic boutons. And basically, there are two types of perisomatic innervating interneurons in the hippocampus, those that express parvalbumin, and as I said, those that express cholecystokinin. But cholecystokinin is not expressed in the terminal, so we cannot use it to identify the terminal. They express CB1 receptor, a cannabinoid receptor. And actually, this is highly expressed in the terminal. So if we look at a Jeffrin punta here, we can say whether this Jeffrin punta is from a CCK basket cell because it expresses this marker, or that other punta, for instance, here is coming from the PV. And when we count that, what we found was a very steep relationship between the number of boutons and the distance to radiarum. So PV basket cell innervate much more deep cell than superficial. And CCK basket cell innervate much more superficial than deep cells. So that tells you that these basket cells that presumably are activated by the entorhinal cortex, they tend to inhibit these pyramidal cells in the superficial layer. Whereas those basket cells, which are more typically activated by the Schaffer, are more typically innervating deep cells and determining that dissection. So you may have two different CA1 cells responding to the different channels of communication, basically because interneuron is silencing them, so that they are more prone to respond to inputs coming from entorhinal cortex of CA3. And actually, when we were working in this data, we knew about the work in the lab of Ivan Soltes, and basically he was recording in vitro from pair recordings in vitro, and he was seeing exactly the same phenomenology of a preferential innervation of PV basket cell for deep CA1 cell. But something that Ivan noted, and that was extraordinary for us, was that he also noted that superficial pyramidal cells preferentially activate PV basket cells as compared with deep. So this tells you not only that you have this deep superficial organization of the innervation of inhibition, but also within columnar. So remember that the hippocampus is the archicortex. Evolutively, it's the first that developed. So we have a primordium here of the neocortex that later evolved evolutively along the animals. So this deep superficial grading of PV-mediated perisomatic inhibition should be important in this conceptualization. And so, as we were discussing, is any other factor? CA3.
Basket cells uniformly distributed across these layers, or you also find them more in the superficial layers?
Well, there is a little bias, but what really determines connectivity seems to be the dendrites, and whether they reach the layers from which the inputs are coming.
Okay, so these basket cells, their dendrites are then uniformly covering the full extent?
Yeah, they are there. They are basically exposed to receive, as we saw in one of the first.
Well, you don't have to go all the way back.
Okay, so basically, yes. One thing is important electrophysiologically to understand is that PV basket cell, they release GABA very synchronously. They are very effective in releasing GABA. Whereas CCK basket cell, they release GABA very asynchronously. So this means that these cells are super efficient in transmitting information, but those may fail. And that is another component of the differences. But here we are saying that not only, that also the. Okay, so any other factor? Let's talk about the microcircuit factors here. This is a UV-GAD Venus A transgenic rat that express GFP, well, Venus A is a kind of variant of GFP, in the, all interneurons. And what you see here, actually, well, this is some marker which is expressed in the nuclei of the pyramidal cell, the red one, it doesn't matter what. But basically, if you look at this innervation occurring here, it's the perisomatic, all these are butons. Butons from the vast majority of interneurons, whatever interneuron you have interneuron. You see your question, Paul, all these cells probably, most of them are basket cells. Well, those are different type of interneurons and here. But well, basically, if we take a look at the perisomatic innervation, we see how dense it is, and it's gaining more and more density as we go here. So this is CA3, and here is CA2. Now, CA2 is a very important region because it has been considered a transitional region between CA3 and CA1. CA2 can be marked by some specific markers like PCP4, alpha-actinin. So we can use immunostaining to identify the cell types. So this cell is a PCP4 positive cell, it's a CA2 cell. Now, there was this data when we were working in this project coming from Tonegawa's lab that using optogenetic and some fancy transgenic mouse they have for expressing channel rhodopsin in this CA2 cell, and they were recording from CA1 where they were illuminating the axons. And what they found was that CA2 axons were more efficient in activating responsive in deep cell than in superficial. So CA2 is projecting more, CA3 is projecting to CA1, both in deep and superficial, but CA2 is projected to CA1 preferentially over deep cells as compared with superficial cells. And actually, there is something which is very, very important, that if you stimulate CA3 and you record from a PCP4 CA2 cell, you see that this cell is rarely can fire. If CA3 is activated, the cell will be very strongly inhibited by feed-forward inhibitory circuits, those perisomatic innervation that I showed you that was so much strong here. And actually, if you record CA2 cells together with the CA1 Sharp wave ripple, you see then at the peak of the Sharp wave, actually those cells, CA2 cells, tend to be silent because, as we said, CA3 start firing and recruit this inhibition and inhibit CA2 cells. So the hippocampal Sharp wave microcircuit, remember we are dissecting the intra-hippocampal circuit from CA3 to CA1, it tells us a lot about how this is organized. CA3 start firing during a Sharp wave and activate all these different cell types. But basically, because CA2 is under the strong inhibitory control of local inhibition, these cells here start firing. And because they innervate much more of deep cell, those cannot respond, they are not receiving the excitatory inputs coming into the basal dendrites together with the excitatory inputs coming into the distal dendrites, because CA2 cells are inhibited due to this strong inhibition. Then, because PV basket cells are more innervating much more deep cells, and they are activated by the Sharp wave, those cells inhibit strongly deep. So there is no way deep cells can be activated efficiently during ripples because they are under a strong inhibitory control here. Whereas superficial cells, they are poorly innervated by PV, they are not too much innervated by C2, they are massively innervated with the Schaffer. Also, they are closer to the border to radiarum, so presumably that distance here is shorter than the distance here, more efficiently integrate, so those cells are more responding to CA3. So this means that the intra-hippocampal information flow is not uniform, and is flowing from the proximal to distal regions and from deep to superficial, and this interaction between these two axes is explaining all this phenomenology on how these cells are recruited.
But the circuit, the way you draw it now, and we would not look at your physiology, would not necessarily account for that, right? Because you just look at the inhibitory interactions here, you would think like, okay, they're sort of both driven by CA3. CA2 is actually not doing anything most of the time.
No, it's releasing. It's doing a lot. It's taking out. If you leave it, it will compete with the CA3 activation, and it will probably make this cell fire.
This diagram, CA3 drives the whole circus.
Yeah,
right?
CA3 start.
It has to kickstart
Yeah.
the whole thing.
Yeah.
If CA3 is driving, CA2 shuts up because of the inhibition.
Mm-hmm.
And then you have a slight difference in the inhibition onto your deep and superficial CA1 cells.
Yeah.
But overall, this only makes sense if there's a very fine balance between the excitatory drive onto these superficial and deep excitatory cells. Otherwise, they would just be inhibited.
Yeah, why don't you think it is not? Actually, these CCK cells, they don't fire during shock wave ripples. This we know because probably these cells are very poorly activated by the CA3 inputs, whereas those are strongly activated by the inputs.
Yeah, but no, it's just a feed-forward circuit, right? So there's no internal.
No. But there is an internal, as I suggest, as I show, the data said that these superficial contact preferentially with these basket cells and inhibit this. So the internal control is not in this cartoon, but yes, it is important, and it also has to be integrated. So,
Just a question.
yes,
Can you go back to the
yes.
So if the CA3 seems to be the only population which is not symmetric in terms of firing rate aligned to the ripple event.
Well, actually, yeah. See, sorry.
Would you comment on that?
Yes, actually, CA3 has some heterogeneous behavior also. So I can talk about that later, but it makes my talk another hour longer. So, but the bottom line
I
is
think she's trying to tell you she doesn't want to talk about it, Diogo.
it's tape recorded. This is published, the other is not. And actually, yes, and actually, bottom line is that this complexity can be understood. So you may feel lost in all these different cell types. Actually, what we have in the lab is a cartoon, and we are bringing, like, you know, the usual suspect. So you're bringing pictures and removing pictures, making links. That's the only way to try to update but at the end, we will emerge with an updated view and updated physiological context to understand that. Yeah. So we said that we were talking about shock waves, and we were saying that these shock waves were actually reflecting this depolarization of the dendrites of CA3. But I also showed that this shock wave is occurring in the radiarum, but the ripple is isolated in the CA1, in the stratus pyramidale. So you may ask why these differences and whether they are reflecting the different components. Actually, that is clearly seen in this laminar recording that you see the shock wave around the electrodes that are recording from the stratus radiarum. And then the ripple, the high frequency components, very stable here around the electrodes which are near this. So what are ripples made from? Now, if we take cells, we look at now, let's take a look not at the depolarization as a global thing, but let's take a look at the structure of the events that accompany each shock wave ripple in the superficial cells and in deep cells. If you are a physiologist and you will have your eye trained to look at, you will notice that these are excitatory components here. Those are EPSPs. So basically, this is the synaptic input from some presynaptic cell activating this particular cell here. And if you take a look here, there is some structure here in this individual event here. So inhibitory components coming here. So if you take the cross-correlation between the field at the stratus pyramidale and this component here, you see that the intracellular have a power in the this band, which is very close to the ripple band. The cross-correlation between these two is somehow, but not that much. But if you look at the cross-correlation between this inhibitory component here and the field, you see that is much more higher. Actually, that tells you that basically deep cells are contributing more to the ripple. And this is what you can use to guess where you are in the stratum. This is basically what the lab of Bouchaki has been doing to localize the position of the electrode within the stratus pyramidale. The power of the ripple along the stratus pyramidale tells you the position, and we could calibrate that because we knew exactly where the cells were. But apart from this mathematical tool, it tells us that there is a larger contribution of deep cell to ripples. Now, remember, superficial were more correlated with the charge wave, and deep are more correlated with the ripple. So the local field potential is not democratic representation of what the different cells are saying. Charge waves are more reflecting the global influences of the superficial cell, and ripples are probably reflecting what is going on at the level of the deep. And actually, that you can understand if you take a close look to tetrode recordings. So we can obtain tetrode recording from the different layers in the rad. We can bring several tetrodes and look at how this ripple looks like. And actually, if you take a close look to a single event, you can clearly see how you see spikes of a presumably pyramidal cell, and some positive components, very clean, where you don't see this firing. And actually, if you look at this firing collapse, meaning that many cells in the neighborhood of this electrode are firing here, but not here. So basically, this means that this should be reflecting firing from different subset of pyramidal cells, not that much because it's not large. I can still identify some. And that is reflecting probably the common inhibitory component. So the building block of ripples are actually composed of these a pyramidal cells and a PV basket cell. So if you take this cell firing, for instance, you activate this cell and it's connected with that, you can see in the field, you have an electrode in the field, you see in the field that inhibitory component as a field component. Basically because this PV basket cell is innervating all of these pyramidal cells together. And because they are releasing GABA simultaneously and inhibiting simultaneously all of them, they will be strongly inhibited together and generate a field. And actually, this is what basically you can see from that build-up of the ripple with the population spike where many cells fire and the inhibitory component. So this pyramidal cell PV basket cell is a basic local motif that is associated with that. Of course, other type of interneurons participate, and that should imprint a unique LFP signature, because if we said that this is so much precise and that there's much more boutons in the deep layers as compared with the superficial layers, if I am bringing this cell fire or another cell here, I may be activating different group of pyramidal cells, and therefore that differences component should be in a way reflecting in how the LFP is. So we try to exploit that.
But does that explain that you only see the shortwave ripples when the animal's standing still?
Yeah, well, this is when the animal stands still, you have a very low release of acetylcholine and other neuromodulators. So that brings all the hippocampus into another state in which they hyperpolarize and they are more ready to be responsible to intra-hippocampal activity. Mm-hmm. Okay?
That would make sense.
Okay. So as I said, that precise dissection between the group of cell and how these ripples are built should imprint unique signature at the local field potential. So this is one tetrode, one channel in the tetrode I showed before. And these are two events which are consecutive. So you see this event and that event. Both are ripples, but they are pretty much different. So probably the cells that are participating here and the cells that are participating here are differently. Probably because it's reflecting that ensemble that has to be replayed. So can we extract that information from all these? Well, I mean, we came across many, many ways on trying to do that, and we were using several unsupervising algorithms. And at the end, we learned from a previous work by the Dragon group who was using self-organizing maps. Basically, this turns to be especially suitable to handle with the singularity of these ripples. So basically, what you do is, so you take, for every event, the 10 first principal components of the LFP, and then you plot them in a 10-dimensional space. So imagine a high-dimensional structure like this, and imagine that different events actually can be plotted here, and we can color label them. So self-organizing maps works to try to reduce dimensionality from this into a 2-dimensional space in which they cluster events that topologically were close in this high-dimensional classification, and they put them together so that we can put together ripples that eventually seems to be more or less classified similarly. So we can think that actually those ripples here were close here in the original space, those ripples here, they are close here, and so on. And if you think that this categorization is reflecting this classification, the fact that one cluster, for instance, reflecting one memory item, is composed of the particular activation of a set of CA3 and CA1 cells with interneurons, that would imprint a particular signature, but the other ensemble should imprint another signature. So we have to see how much of them can be flipped between this and that, how close they are, or how different they are. And this gives you a whole number of events that can be classified. Now, we apply this to our data. So from 325 events that you have, we asked them to be classified, and they were classified in a number of motifs. Of course, we can discuss on the limit of this classification, whether it's meaningful, it's not meaningful, but for us, all this discussion ends when we saw how was the firing rate of a single cell during that classification, which was unsupervised. So basically, what we saw was that when we led the algorithm to classify, and then we asked what was the firing rate of that cell during those events in these juxtacellular recordings we were doing, what we found was that this particular cell had a preference to fire around these ripples, but not around those ones. And actually, you can define clusters here based in the topological similarities between the events being classified together. And you can run a statistical test and see whether the participation is different than chance. And basically, this tells you that it is different, that the cell is preferring, this particular cell can prefer to fire here.
Liset, wait, now I'm getting a bit confused because we have the ripples. The ripples are a circuit phenomenon. It's the cell that's part of these circuits. And now you're saying, well, cells are not linked to all the short-wave ripple circuits. They have a preference for certain short-wave ripple circuits.
Yeah.
Right? Is that amazing?
No, it is not.
Ah, okay. Okay, this is just to establish that fact. You say, look.
Well, I mean, you know, this selective reactivation that you have for the different. So I started my talk
We
with
go
this.
back to burger.
No, well, basically, if you.
Sorry, how much time do you have? Because it's.
So 15 minutes? That's okay?
Yeah, that's
Otherwise,
perfect.
they don't give us lunch.
Yeah, no, wait for us, please.
Liset will buy everyone lunch when it's
I
finished.
will not have lunch. Okay. Thank you for your patience anyway. So that selectivity in the way in which they are replayed should be retained somehow.
Okay, so
That's ugly. That's ugly. Okay. We are coming to the end, but don't worry. So as I said, so the participation is not random. The single cell chooses to fire at a particular type of events and not the others. And actually, there is this difference between deep and superficial in how selective they are and the participation to the different clusters. You can imagine that a superficial cell will be more firing more to different ripples, probably, as a deep cell that shouldn't be more selected, actually. So that single cell firing preference by ripple pattern is meaningful, and the differences between deep and superficial cell that we were explaining before can be explaining in terms of how much of the inhibition, how much the different cells are connected by interneurons so that they become more or less selective of the different ripple patterns. So superficial cells become more frequently engaged of these differences. So what determines that selectivity? And here is where it is important to remember all these discussions we were having, whether a cell was following this drive with the global drive at the stratum radiarum, so that they follow more with the sharp wave sink or not. And basically, if you run the same analysis from the intracellular data, you look at the firing preference. Again, you see the cell was specific of these events here. And here is where the driving force of the cell was more strongly depolarized, for instance. So this means that the cell fire here because it's more depolarized there. However, if you look at the simultaneous matrix of the differences sink associated with all these clusters, you see that that matrix and that matrix poorly correlate. Actually, this matrix correlate pretty well with the matrix of the CA3 multi-unit firing. So this means that this global signal is not following by this particular cell closely, because actually this cell was deep cell. But a superficial cell will follow more closely that. So that difference between the group and the individual is given the different cell types in this superficial the ability to do something which is not as expected. So they should not fire probably at the theta peak because of the position. It's making this cell more selective to that. Actually, these deep and superficial differences are now becoming hot topic, and many labs are starting to see differences. For instance, the lab of Sebastian Royer using silicon probe and animals running in this treadmill with landmarks, somatosensory landmark, what he found was that deep cells were more typically anchored to somatosensory landmarks in the treadmill, as compared with superficial cells that apparently reflect other kind of spatial representation. Previously, actually, in 2013, when we were studying somatosensory activation of the hippocampus, we stimulate the hind paw, the forepaw, and the whisker. We were recording with tetros in the hippocampus, and we noted some CA1 cells being inhibited, some other cells being depolarized. Well, by that time, we were not being able to identify the deep and superficial nature of those cells. So the lab of Attila Losonsi is using GCAM indicators, and what they have found was that deep cells has a more spatial place. There are more place cell fraction in deep cells than in superficial, and this goes very well a match with electrophysiological recording from Mizuseki in Bushaki's lab, who was the first who started talking about these differences between deep and superficial. In this case, he found only differences between theta events during REM sleep as compared with exploratory theta. In that particular paper, only that difference were highlighted. So deep cells may obey to somatosensory landmarks. Larger place cells fraction may be in the deep superficial layers. And to finish and to try to come up with a clear final message, and going back to how we start discussion, deep cells are more tuned to entorhinal inputs than superficial cells. So actually, those are recording along the theta cycles. And if we decompose the recordings into a conductance components that is variable along the theta phase and in the driving force that we were discussing before. And what you find was that for deep cells, actually, at the phase, at the theta peak, when the entorhinal inputs are expecting, there is a much more depolarizing current flowing through deep cells as compared with superficial cells at the phase at the theta peak. So this means that deep cells are more like to be activated by entorhinal cortex as compared with deep cells. And in contrast, during the late phases of the theta oscillation, what you have is more alignment of this depolarization in superficial with this with the income, with the arrival of the CA3 inputs, as compared with deep cells at this particular phase. Then big cells fire more at the theta throw because there is this rebound of depolarization there in which the cells are more likely to be tuned to fire. But if you put the preferent theta phase and you cross-correlate this with the distance to radiatum, what you have is a cross-correlation, although you see that for deep cells, you have a degree of variability that we still need to understand. So that explains basically why the deep and the superficial could actually participate differently in this phenomenology of deep cell to be more responsive along different phases of the theta rhythm. So to finish, the entorhinal hippocampal system is much more complex, and these three synaptic circuits is even more that we originally thought, and actually we still need to put labels here, make wires here, and try to understand how these are organized. And before finishing, let me just take 2 minutes, just 2 minutes, to tell you something about what also we can learn from neurological disease. We have heard about HM patients who have told us so much about the role of hippocampus in memory. But temporal lobe epilepsy is a disease that affects temporal lobe hippocampal circuits specifically, and this can be modeled in rodents. And this is work by Neil Burgess, actually, showing that what we have been conceptualizing about what's the role in the hippocampus in memory is much more complex than that picture we emerge with from HM patients. And in the human, there is a strong laterality of the function, cognitive function. So people having left temporal epilepsy, they, when they navigate in a virtual maze and they are asked to localize objects or to remember places with objects with episodes that happen in the field, in the virtual maze, people with left temporal lobe epilepsy, they are poor in episodic memory, but they can perform pretty similar to control in object recognitions. However, people having right temporal lobe epilepsy, they are pretty much okay with episodic memory, but they can fail in the object recognition task. So spatial recognition memory and episodic memory function are dissociated. In the case of human, it's a strong laterality, but we still don't understand how these episodic-like memory deficits may be explained by a specific circuit alteration. And all the work we have been done in the lab for the last year was trying to look at how these different oscillations are altered in normal and epileptic rats, and how from these different alterations in the theta, gamma, and ripple band, we can relate with the behavior of the animal in memory tasks. And we have seen that epileptic animals, when you do this carefully, they show deficits in what, where, when memory tasks, which is a kind of episodic-like memory task, but not in the Morris water maze and not in object recognition tasks. So eventually, that new perspective of hippocampal function should be also translated to this function to try to understand all these problems at a more elaborated level. And just to convince you that there is a strong deep superficial regionalization, these are RNA-seq data obtained by isolated superficial and deep layers. And what we found was that in epileptic rat, from all the genes that are upregulated, 200 E genes are upregulated specifically in superficial layers. That genes affect synaptic formation, axonal wiring, and ionic channels and report. So we still need to understand a lot about how hippocampus is organized in the rodent and the human, and how does it relate to cognition. So to summarize, the functional organization of the hippocampus is far more complex than the classical three synaptic circuit today. In CA1, a deep superficial axis interacts with the proximodistal and the dorsoventral directions, and dedicated ensembles establish cell type specific links within the entorhinal hippocampal networks. So I want to thank people in my lab, funding agency for their support. All the people have done the work, and we are always looking for talented students who wants to join us to try to understand these questions at the multicellular, multiple levels of complexity. Thank you.
Okay, fantastic. We started today believing the hippocampus was simple, but I think we can throw that idea out of the window now. So who has questions for Liset? So Liset, so in some sense, we get a complexification now, right? So we had sort of, there's a simple idea about how these circuits work, and now you start to add a lot of, let's say, variability, dynamics. But what are the, if you look at this from a modeling perspective, which means you want to abstract, you want to really focus on specific principles, ignoring a bit the variability, what are the principles do you think any decent model of the hippocampus should satisfy? So let's look at that Niels' proposal.
Yes.
Which features should it
I think
incorporate?
you should understand that the code is at the population, is at the ensemble. It's an ensemble code, and that you need specific players for signaling the vast majority of complexity that you can determine. And so when you wire a grid cell with a CA1 cell, you need probably to try to implement somehow how this wiring occurs, because we still don't know precisely how this layer 3, which, by the way, grid cells expressed along the layers, is something that we still need to clarify much more, and how this particular positioning of the particular cells are connected into this circuit, because this is an auto-organizative network with attractor dynamics. That network is organized in a feed-forward manner. That network here is designed for pattern separation. So the way in which all these components connect to those networks is meaningful for how the outcome. So my bottom line is to learn more from this dedicated microcircuit connectivity and to implement that at the level of.
Okay, but for instance, now, look, so you were just summarizing your results with respect to CA1, where you say, well, entorhinal cortex has a differential effect on the superficial and deep layers in CA1, right?
Yeah.
But now you see here at entorhinal cortex that, again, there's quite some spatial structure to it. So
Yes.
for instance, does medial and lateral entorhinal cortex also drive then that CA1 field differentially?
Well, in the proximal to distal, there are differences. So this means that the grid cell will fit more in the proximal than in the distal, whereas border cells and object border cells will fit more in the distal than in the proximal. So the complementary code will emerge in the network, the way in which they are connected.
Yeah, but in the meantime, lateral and medial will also directly drive dentate.
Yeah, well,
and they start to mix,
yes,
right?
of course,
and then
of
also
course.
drive C1.
Yes,
so
of course.
how should I think about that?
We should need more about how it is connected and try to identify simple rules. But as I said, I think that mathematically, it should be the way in which you integrate activity from grid cells here at the proximal, or activity from object cells more here at the distal, should have an impact on how the whole system code for demand.
Okay. Yeah, so thanks for the great talk. I think finally I understood like some of the mysteries about like phase amplitude coupling.
You will give a lecture about it this afternoon.
No, I don't think of it. But missed chance. But the point is, you see phase amplitude coupling and theta gamma coupling not just in the hippocampus.
Yeah.
It's a phenomenon that also happens, for example, between anterior cingulate cortex and medial prefrontal cortex. So now that you defined like this circuitry, like so specifically for the hippocampus, like to determine how this phenomenon then is created, would you argue for like a similar type of circuitry, like in other cortical areas, or would you argue for like, I don't know, something different that leads to the same oscillatory phenomena?
Yeah.
Like, how would you interpret this? Like?
So there are general microcircuit motifs that repeat in all the areas. So pyramidal cell and interneuron, which is PB basket cell, is present in the hippocampus, in the neocortex. The important is what inputs are activating them, whether they are feed-forward activated by the thalamus or by the collateral from the contralateral cortex,
Right.
or whether they are more feedback activated by the pyramidal cells. So pyramidal cell interneurons is a basic motif,
Sure.
which is basically the most elementary circuits underlying gamma. There is, that's the reason why gamma is so much conspicuous. But the way in which it is organized for with the inputs depend on the structure, and you need to know.
Yeah, it's beautiful to see all the differentiation between deep and superficial, yet it also is a bit puzzling. Because if you, for instance, describe how the deep cells receive more entorhinal input, possibly about sensory information and landmarks, would seem important to integrate that with space, for instance, with spatial. And how does that happen once you segregate into these two layers?
Yes, yes. Well, actually, you see this data emerging from GCAM animals that you have different representations. So this deep and superficial matters. But you also have to know, as I said, that the proximodistal axis determine whether the medial entorhinal cortex project to the mostly to the proximal, and the lateral entorhinal cortex project to distal. So as I said, is this interaction between the deep superficial and the proximodistal what matters. And again, that depends on what process are you taking a look at. So if you are in a theta, the rat is engaged in object exploration, there is a massive theta activity and a dominance of the entorhinal inputs, which is more probably stronger at that level than the intrahippocampal loop. So although that intrahippocampal loop may determine whether from CA3 to CA1 is firing or not, they should be obeying more to those inputs coming from the entorhinal cortex. Now, because the deep and superficial are also regulated by the interneurons, not only feed-forwardly, but also by feedback inhibition, that would determine whether one cell will jump in advance into the phase or not. For that, you need to dissect the inputs coming to that particular cells. We are trying to do that with rabies viruses, for instance, to try to determine whether a single cell that we can record and see whether this single cell is obeying to more entorhinal that CA1, CA3 inputs is more connected or less connected with. Because at the end, the ensembles matters.
Yeah, you also need to know what comes in at the distal dendrites in terms of entorhinal.
I was wondering if you, in all this complex circuit you describe, if you, in general, agree with this idea of CA1 as receiving inputs from CA3 or entorhinal for encoding and retrieval phase memory processes. Okay. And in the deep superficial axis you describe, how encoding and retrieval will take place.
Yeah, this has to change dynamically. Okay, so there is no, whether there is a circuit for encoding, which is separated for a circuit for recalling or that retrieving, that is not the case. I mean, it's dynamically involved in how the circuits are. But now remember that the cell will fire along the theta phases, and they will find, if you speak to me now, you will try to communicate with me in a moment that some people are talking to me. So I will probably not be able to attend to you because I am so much engaged in this conversation that I will not be able to pay attention to that. But if you speak to me out of phase, I will probably communicate very well with you at that moment, and we will establish some links. And eventually, once we establish some links, if I am talking again here and you now communicate with me, I will probably turn my head and talk to you because our links are stronger. So the way in which the links are established by these synaptic plasticity mechanisms depend on how the different cells are firing. And that's occurred by this modulation along the cycles. So to put that picture on top of that picture requires to integrate this in a dynamical model of how these evolve when the animal is moving around the field.
So I have a very brief comment because I'm hungry. But we recorded, if you record as rats run around in a novel environment or a familiar environment, you see a shift in the preferred phase of firing in the appropriate way, as if the novelty should be more entorhinal driven. And it's blocked by scopolamine. So it seems like Mike Hasselmo's proposal in the 1990s that the phase of theta should relate to novelty or familiarity and be cholinergic has some strength.
Yeah, well, that we can discuss later if you like, not to retain people here. But yes, there are many differences in the expression of modulators, receptors to acetylcholine and other neuromodulators between these layers, but also how these two cell types project to the amygdala and to the prefrontal cortex and how they are engaged with it. So probably novelty is a major trigger to reorganize how the circuit is conveyed.
Fantastic.
So
Okay,
thank you,
Liset, thank you very much for a great talk.